The blood sample arrived on a Tuesday. I remember because Tuesdays were when we processed routine wildlife DNA extractions: black bears, cougars, the occasional wolf from reintroduction monitoring, standard conservation genetics work. Nothing that would change the trajectory of my career, my ethics, or my understanding of what we owe to the hidden corners of the natural world. The sample came in a standard cryogenic vial, labeled only with coordinates and a collection date, March 17, 2018, from the Mount Hood Wilderness. The submission form listed it as unidentified primate tissue, possible contamination event. A ranger had found it on a tree branch at 7,000 feet, mixed with fur and what appeared to be dried blood from a territorial marking behavior. My name is Dr. Rebecca Torres. I'm a conservation geneticist, or I was before I became a footnote in a classified file. For 6 years, I worked at a joint USDA Forest Service and National Institutes of Health research facility studying disease resistance in wildlife populations. My specialty was comparative immunogenetics, understanding how wild animals survive pathogens that would kill their domestic relatives. I extracted the DNA on a Wednesday. By Friday, 3 men in suits were standing in my lab. By Monday, my research no longer belonged to me. The facility occupied a converted fire station in the Gifford-Pinchot National Forest, 40 miles from the nearest town. We were a small operation, 4 research scientists, 2 lab technicians, and a rotating cast of graduate students who came for the fieldwork and stayed for the isolation. The location was deliberate, close enough to wilderness areas for sample collection, remote enough to maintain biosecurity protocols for the pathogen work. My project focused on disease resistance in wild canids and ursids. Specifically, I was trying to understand why certain populations of black bears showed remarkable resistance to parasitic infections that devastated others. The hypothesis involved major histocompatibility complex diversity, the genes that govern immune response and tissue rejection. If we could identify the genetic markers for resistance, we might be able to predict which populations would survive as climate change pushed parasites into new territories. The work was meticulous and unglamorous. I spent my days extracting DNA from hair samples and fecal matter, running PCR amplifications, sequencing genetic markers, and building databases of genetic diversity across populations. Most samples yielded exactly what we expected: standard ursid DNA with minor variations in immune-related genes. The sample from Mount Hood was different from the first analysis. Initial sequencing suggested primate DNA, which wasn't impossible. Contamination from the ranger who collected it, perhaps, or from one of us in the lab, but the quality was too high for surface contamination. This was tissue-level DNA, intact and abundant, from something that had left significant biological material on that tree branch. I ran it through GenBank, the NIH's genetic sequence database. No match to any known primate. I tried more specific databases: great ape genomes, New World monkeys, even fossil DNA from extinct hominids. Nothing aligned properly. The sequences showed clear primate characteristics: the overall genome organization, the chromosome structure, the presence of genes that exist only in primates. But the specific base pair sequences diverged from human DNA by approximately 1.2%. For context, humans and chimpanzees share 98.8% of their DNA. This was something phylogenetically positioned between modern humans and our extinct cousins, but distinct from all known lineages. I thought it was contamination, a database error, maybe degraded DNA creating false readings. I extracted again from the original sample, using different protocols, different equipment, even different reagents to eliminate any possibility of laboratory artifacts. Same results. I called Dr. James Koh, my supervisor, into the lab. Jim was 63, Korean American, with a dry sense of humor and an encyclopedic knowledge of mammalian genetics. He'd spent 30 years with the Forest Service before transitioning to research. He'd seen everything: poached grizzlies, hybrid wolves, escaped exotic pets causing genetic chaos in wild populations. He looked at my sequencing data for 20 minutes without speaking. Finally, run the mitochondrial DNA, full sequence. Mitochondrial DNA is inherited maternally and evolves slowly, making it ideal for determining evolutionary relationships. It took 3 days to sequence the complete mitochondrial genome from the sample. The results placed it firmly within the Hominidae family: humans, chimps, gorillas, orangutans. But it didn't match any known species. The divergence pattern suggested it had split from the human lineage somewhere between 500,000 and 1.5 million years ago. roughly the same time period when Homo erectus was spreading across Asia and Europe. This is either the most significant find in paleoanthropology in 50 years, Jim said quietly, or we're about to become a cautionary tale about laboratory contamination. The tissue sample is intact, I said. Fresh. Less than a month old when it was collected. This isn't fossil DNA. Jim looked at me with an expression I couldn't read. Then we have a problem. He picked up the phone and called someone at NIH headquarters. That's when everything changed. The 3 men arrived 4 days later in an unmarked SUV. 2 were from the Department of Defense. They introduced themselves as contractors working on biological security assessment. The third was from the NIH, from a division I'd never heard of, the Special Pathogens Branch. They wore suits poorly suited to the forest environment and carried briefcases that seemed too heavy for documents alone. They weren't interested in chit-chat. Dr. Torres, we need to discuss your recent sequencing results, the NIH representative said. His name was Dr. Raymond Chen, and he had the exhausted look of someone who'd spent too many years managing crises. Specifically, the unidentified primate sample from the Mount Hood collection. It's a remarkable find, I said carefully. If authentic, it represents... We know what it represents. One of the DoD contractors, he'd introduced himself only as Miller, opened his briefcase and removed a tablet. This isn't the first sample we've encountered. It's the first that's been properly sequenced. He showed me a map on the tablet screen. The Pacific Northwest, dotted with red markers. 57 locations, scattered across Washington, Oregon, and Northern California, all in remote wilderness areas above 4,000 feet. Each marker represented what he called an organic material recovery. Tissue samples, hair, blood, fecal matter. We've been collecting these for 6 years, Dr. Chen said, as part of a multi-agency effort to assess potential zoonotic disease threats in wilderness areas. Most samples were too degraded or contaminated for analysis. Yours is the first complete genome sequence we've obtained. Who else knows about this? Miller asked. Just Dr. Coe and myself, and now you. Keep it that way. This work is being reclassified under National Security Directive 31, which governs research into potential biological weapons and novel pathogens. Your facility is now operating under enhanced security protocols. I felt something cold settle in my chest. This isn't a pathogen. It's an organism, possibly a surviving population of an archaic hominid species, which represents a significant national security concern. Miller interrupted. Unknown primate populations in domestic territory, with unclear disease profiles, unknown behavioral patterns, and now confirmed genetic distinctiveness. The implications for public health, conservation policy, and territorial security require immediate assessment and containment. Containment? I looked at Dr. Coe, but he was staring at the floor. Dr. Chen's expression softened slightly. Dr. Torres, we're not the enemy here. We need your expertise. But this discovery cannot become public until we understand what we're dealing with. The scientific community, the media, the public, none of them are prepared for this information. The chaos alone would be catastrophic. They gave me a choice. Continue the research under their supervision, with full access to their sample archive and unlimited resources, or be removed from the project entirely. The research would continue regardless. The only question was whether I'd be part of it. I thought about the sequence data, about the implications of a living hominid population that had survived, hidden, for hundreds of thousands of years, about what might be learned from studying their genetics, their immunity, their adaptation to environments humans found uninhabitable. I thought about the word containment. I'll continue, I said, but I need full disclosure about what you've already found. All samples, all data, all previous research attempts. Miller exchanged glances with Dr. Chen. Agreed. But understand, from this moment forward, nothing leaves this facility without clearance. No publications, no conference presentations, no casual conversations with colleagues. You're now working on a classified project. He had me sign 17 pages of non-disclosure agreements, security protocols, and legal liability waivers. I signed them all. They brought me 43 additional samples over the next 2 weeks. Each arrived in a locked transport case hand-delivered by military courier, with chain of custody documentation that read like something from a criminal investigation. The samples varied: tissue, hair, blood, even what appeared to be skin cells scraped from tree bark. Some were fresh, collected within the previous month. Others had been in frozen storage for years, waiting for technology sophisticated enough to analyze them. I worked 16-hour days, running extractions and sequences, building a genetic database that no one outside our facility would ever see. Dr. Ko supervised, but the real oversight came from Dr. Chen, who appeared every 3 days to review results and ask increasingly specific questions about immune function, disease resistance, and genetic compatibility with human cell lines. That last question should have warned me. The genome that emerged from the analysis was extraordinary. These weren't just surviving hominids. They were adapted in ways that suggested hundreds of thousands of years of evolution in harsh, isolated environments. Their immune genes showed remarkable diversity, with allelic variations I'd never seen in any primate population. The major histocompatibility complex was particularly striking. They possessed immune receptor variants that appeared capable of recognizing and responding to a broader range of pathogens than human immune systems could manage. But it was the regenerative markers that changed everything. I found them by accident while analyzing cellular senescence genes, the genetic mechanisms that control aging and cell death. In humans, these genes activate after a certain number of cell divisions, triggering aging and eventually death. They're a fundamental constraint on human lifespan and healing capacity. In the unknown hominid samples, these genes were different, not absent. These weren't immortal cells, but modified, with regulatory sequences that suggested dramatically extended cellular lifespans and enhanced repair mechanisms. I ran comparative analyses against known longevity genes in naked mole rats, bowhead whales, and other species that show unusual aging resistance. The hominid genes were more sophisticated. I called Dr. Chen immediately. He arrived within 6 hours, bringing Miller and a new face, Dr. Sarah Vasquez, who introduced herself as a regenerative medicine specialist from Walter Reed Medical Center. She looked at my data for less than 5 minutes before her expression changed from skepticism to something approaching awe. These regulatory sequences, she said, pointing to a section of the genome. If they function the way this suggests, we're looking at cellular repair mechanisms that could revolutionize treatment for degenerative diseases, traumatic injuries, maybe even cancer. You're talking about human applications, I said. I'm talking about saving lives. She pulled up additional screens on her laptop. Do you know how many soldiers come back from deployment with traumatic brain injuries? How many people die from organ failure because regeneration capacity is too limited? How many children suffer from genetic conditions that prevent normal cellular repair? Dr. Chen held up a hand. Dr. Torres, what we're about to discuss is classified at the highest level. Before we continue, I need your confirmation that you understand the implications of this work. You want to use their DNA to develop medical treatments. We want to understand if cross-species genetic therapy is viable, if these regenerative mechanisms can be isolated, synthesized, and potentially introduced into human cell lines for therapeutic purposes. It's still theoretical, but if successful, we could develop treatments for conditions that are currently death sentences. I looked at the sequence data on my screen, at the genetic information extracted from blood left on a tree branch by something that had probably been marking territory, communicating with others of its kind, living a life that had nothing to do with human medicine or military applications. Have you considered, I said quietly, that this organism might not consent to being used this way? The room went very silent. Dr. Vasquez left the next morning, taking copies of my genetic sequences and a sample of extracted DNA. She promised to have preliminary compatibility studies completed within 3 weeks. Dr. Chen stayed behind to oversee what he called protocol enhancement, which meant turning my research facility into something resembling a military installation. New security systems were installed, biometric locks, surveillance cameras, encrypted communication networks. 2 armed security personnel took up residence in a trailer outside the facility. All biological samples were moved to a new secure freezer that required 3 separate authorization codes to access. My personal laptop was confiscated and replaced with a government-issued device that logged every keystroke. I was allowed to continue my work, but now I had watchers. Miller sat in my lab most days, saying little, but his presence was a constant reminder that this was no longer pure research. This was something else, something with applications and implications I was only beginning to understand. I started asking questions. Where did the samples come from? Specifically, Dr. Chen showed me the collection data. Most samples had been recovered near trail systems or wilderness boundaries, places where human and unknown hominid territories overlapped. Several came from areas near hiking accidents or unexplained disappearances. A few had been collected after what were officially logged as bear attacks, though the tissue evidence suggested something bipedal with different dentition patterns. Are they hostile? I asked. They're reclusive. Most evidence suggests active avoidance of human contact. But there have been incidents, territorial displays, aggressive vocalizations, occasional property destruction at remote cabins. Nothing that rises to the level of sustained violence, but enough to suggest they're aware of human encroachment and not particularly pleased about it. So we're taking genetic material from an endangered population that's trying to avoid us, to develop medical treatments they'll never benefit from. We're attempting to save human lives using naturally occurring genetic information. It's no different from deriving antibiotics from soil bacteria or cancer treatments from deep-sea organisms. Those organisms aren't sentient primates. Dr. Chen's expression hardened. We don't know what they are, Dr. Torres. That's the point of your research. And until we have clear evidence of sophisticated cognition, tool use, and cultural complexity, we're treating this as a conservation genetics issue with potential medical applications. But I'd seen the genome. I'd analyzed the neurological development genes, the FOXP2 variations associated with language capability, the prefrontal cortex development markers that suggested advanced cognitive function. These weren't simple primates. They were something much closer to us, close enough that the ethical frameworks we'd developed for animal research felt inadequate and possibly irrelevant. I requested access to behavioral data, surveillance footage, if it existed, vocalization recordings, anything that might indicate cognitive sophistication and therefore require enhanced ethical consideration. Miller denied the request. Behavioral assessment isn't part of your mandate. Focus on the genetics. Genetic analysis requires context. If these organisms possess human-level cognition, they don't. Miller's tone was flat, final, and speculation in that direction is unhelpful and potentially counterproductive to the mission. I went to Dr. Coe that night. He was in his office, working late as always, surrounded by years of accumulated research papers and field guides. He looked older than he had a month ago, worn down by something he wouldn't name. Jim, this feels wrong. He didn't look up. Most important work does eventually. They're isolating me from contextual data, making me treat this like it's bacteria instead of what, Rebecca? What do you think they are? I think they're close enough to human that what we're doing crosses ethical lines we've spent decades establishing. He finally met my eyes. Then you have a choice. Walk away knowing the work continues without you, or stay and try to ensure it's done with at least some measure of scientific integrity and ethical consideration. That's not a real choice. It's the only one you have. Dr. Vasquez returned in early May with results that changed the entire trajectory of the project. The compatibility studies had been successful, not just successful, revolutionary. She'd managed to introduce modified versions of the hominid regenerative genes into human cell lines using CRISPR-based gene editing. The results showed dramatic improvements in cellular repair mechanisms, resistance to oxidative stress, and recovery from induced damage. We tested it on human neural cells subjected to traumatic injury analogs, she explained, pulling up microscopy images on her laptop. Control cells showed typical damage patterns: cell death, limited repair, permanent functional loss. Cells with the introduced genetic modification showed repair rates 3 to 4 times faster, with significantly reduced permanent damage. The implications were staggering. Traumatic brain injuries, spinal cord damage, degenerative neural diseases, conditions that currently meant permanent disability or death might become treatable, perhaps even reversible. We want to move to animal trials, Dr. Vasquez said. Mice first, then primates, then potentially human trials within 18 months if safety profiles hold. 18 months? I looked at Dr. Chen. That's impossibly fast for gene therapy development. We have certain advantages, he said carefully. Streamlined approval processes for treatments with national security applications, and significant funding from DoD medical research divisions. You're talking about turning this into military medicine. I'm talking about treating wounded soldiers who currently face lifelong disability, about giving field medics tools to prevent traumatic injuries from becoming permanent catastrophes. Yes, military applications are a priority, but civilian applications follow immediately. This isn't about weaponization, it's about healing. I looked at the data again, at the cellular repair rates, the reduction in permanent damage, the possibility of restoration where previously there was only loss. I thought about my cousin Maria, who'd spent the last 6 years in a wheelchair after a car accident severed her spinal cord, about my graduate advisor, dying slowly from ALS as his motor neurons deteriorated, about the millions of people suffering from conditions that this research might actually help. And then I thought about the source. How many samples do you need? I asked. For initial trials? What you've already provided is sufficient. For scaled development and production, we'd need significantly more genetic material to fully characterize the mechanisms and develop stable therapeutic constructs. So you'd need to go back, collect more samples from the source population. Eventually, yes. And if the source population is small, if increased collection puts them at risk? Dr. Vasquez's expression shifted, becoming less certain. That would be unfortunate. But population assessment isn't my area. Conservation considerations would be handled by Forest Service Wildlife Management Divisions. I turned to Dr. Chen. What's the estimated population size? That's classified. Give me a range. He was quiet for a long moment. Based on genetic diversity analysis, distribution patterns, and collection site density, we estimate between 300 and 800 individuals across the Pacific Northwest. Possibly fewer. 800 individuals. A population smaller than many endangered species. Smaller than the remaining populations of mountain gorillas, Sumatran orangutans, or Javan rhinos. And we were discussing harvesting their genetic material for medical applications that would benefit the very species encroaching on their habitat. This is extraction, I said. We're mining them, taking their biological resources without consent, without consideration for their own survival, to solve problems we created. We're using naturally occurring genetic information, Dr. Chen began, from a sentient species that's been avoiding us for thousands of years. That's hidden itself so successfully we didn't even know it existed until accidentally stumbling onto their DNA. And now that we've found them, our first instinct is to determine how they can serve us. Miller stood. Dr. Torres, I understand your concerns, but let me be clear about priorities. Human welfare comes first. American citizen welfare comes first. Military readiness comes first. If this population possesses genetic information that can save American lives, we have a moral obligation to pursue it. Even if pursuing it destroys them? Species go extinct. It's unfortunate but inevitable. What matters is what we preserve and learn before that happens. The room felt very cold. The project expanded rapidly after Dr. Vasquez's successful compatibility studies. New researchers arrived. Molecular biologists, gene therapy specialists, bioethicists whose jobs seemed to be rationalizing decisions already made. The facility grew. Temporary structures housing additional laboratories, animal testing facilities, secure communication centers. What had been a quiet forest research station became a complex of military-funded medical research that operated under classification so tight that most people working there didn't know what their colleagues were doing. I was moved to a more specialized role, genetic mapping and mechanism characterization. My job was to identify exactly which genetic sequences controlled the regenerative capabilities and how they could be isolated, modified, and introduced into human genomes without triggering rejection or unintended effects. The work was fascinating. Scientifically, it was some of the most sophisticated research I'd ever been part of. The hominid genome contained regulatory mechanisms unlike anything in current medical literature. Genetic switches that activated repair pathways in response to cellular damage. Telomere maintenance systems that extended cellular lifespans without triggering cancer. Immune modulators that prevented both infection and autoimmune response. We weren't just looking at better healing. We were looking at fundamental changes to human biological limitations. But the cost of that knowledge became clearer as summer progressed. Collection teams were deployed into the field. Special operations personnel trained in wilderness tracking and biological asset recovery. They worked at night, using thermal imaging and acoustic monitoring to locate source populations. The samples they brought back were no longer opportunistic findings, bark scrapings, shed hair, abandoned materials. They were targeted extractions, tissue samples taken from living subjects. I saw the methodology reports, dart-based tranquilizers from distance, rapid sample collection during the sedation window, immediate retreat before the subject regained consciousness. The teams were professional, minimizing distress and physical harm, but the fundamental nature of the work had changed. We were hunting them. The subjects show no long-term adverse effects, Dr. Chen assured me, showing me the follow-up monitoring data. We tag them with subcutaneous trackers, monitor their behavior patterns afterward. Normal activity resumes within 24 hours. They experience brief confusion, but no lasting trauma. You're tracking them. You're treating them like research animals. We're treating them like valuable biological resources that require careful management. They're not resources. They're, what? People? Miller leaned against the lab bench, his expression hard. Show me tool use. Show me fire. Show me art. Burial practices, symbolic thought. Show me anything that elevates them beyond very intelligent primates, and maybe we can have a conversation about rights and personhood. Until then, they're wildlife on federal land, which gives us jurisdictional authority for research purposes. I pulled up files I'd been compiling quietly over the past weeks, genetic analyses I'd run without authorization, using equipment time that wasn't monitored as carefully as it should have been. FOXP2 gene variants associated with linguistic capability, present and sophisticated, prefrontal cortex development genes comparable to modern humans, social learning markers, advanced, neural plasticity indicators, extensive. Neurological structure analysis suggests cognitive capability within the range of human variation. I looked at Miller directly. They're not wildlife. They're people. Archaic people. Divergent people. But people nonetheless. And what we're doing to them is a crime. The lab went silent. Dr. Vasquez was staring at my screen, her expression conflicted. Dr. Chen had gone pale. Miller's face showed nothing at all. This analysis, Miller said slowly, is outside your approved research parameters. It represents a significant security breach and misuse of classified resources. It represents due diligence. You can't develop ethical oversight without understanding what you're working with. Ethics are determined by the project leadership, not individual researchers with personal objections. Ethics are determined by facts, and the facts say we're experimenting on people without consent. Miller collected my files, all of them, including my personal research notes. You're removed from the project effective immediately. Your security clearance is revoked. You'll remain in restricted quarters until transport can be arranged. I was escorted to a small room in the administrative building. Not quite a cell, but not quite freedom either. Through the window, I watched as they brought in another sample from the field. I was held at the facility for 11 days. They called it administrative processing, but it was detention. My phone was confiscated. My computer access was revoked. I was allowed outside only for supervised exercise periods, always watched by security personnel. Dr. Co visited once, bringing books and looking more exhausted than I'd ever seen him. They're moving forward with primate trials, he said quietly. Rhesus macaques, modified with the regenerative gene constructs. Early results are promising. Jim, you know this is wrong. I know it's complicated. I know people are suffering who might be helped. I know that knowledge once discovered doesn't go away just because we don't like how it was obtained. That's the rationalization every unethical researcher has used. We're learning important things. We're helping people. The ends justify the means. He was quiet for a long time. What do you want me to do, Rebecca? Walk away? The research continues regardless. At least with me there's someone who gives a damn about doing it carefully. Be a witness, I said. Document everything. Keep records they don't control, because eventually this becomes public. And when it does, someone needs to be able to testify about what actually happened here. He nodded slowly. They're releasing you tomorrow. Back to civilian life. You'll sign additional non-disclosure agreements, accept a generous research grant to pursue independent work, and never speak about what you learned here. And if I refuse, they'll make your life very difficult, revoke your credentials, blacklist you from federal research, possibly pursue legal action for security breaches. They're giving you a way out that preserves your career. I suggest you take it. That night I dreamed about the forest. Not the manicured wilderness near the facility, but true forest. Deep, old, untouched. I walked through trees so tall they disappeared into mist following something I couldn't quite see. A presence moving parallel to my path, keeping pace but never revealing itself. In the dream, I understood. They knew we were there. They'd always known. They'd watched us build our facility, set up our monitoring equipment, intrude deeper into their territory with our roads and trails and certainty. They'd hidden, retreated, adapted their movements to avoid contact, until we'd started taking their blood. I woke to someone shaking my shoulder. One of the security guards, his expression urgent. Dr. Torres, you need to come with me. Quietly. He led me through empty corridors to a service exit I hadn't known existed. Dr. Ko was waiting outside next to a civilian truck I didn't recognize. Get in, he said. Jim, what? They're bringing in more samples tonight. Larger extractions. They've located what they think is a family group, juveniles present. They're planning to take samples from all of them, including tissue biopsies that require more invasive procedures. From children. From juveniles. Whatever you want to call them. He handed me a hard drive. This contains everything. All the genetic data, all the collection reports, all the behavioral assessments they said didn't exist. Video footage of the darting operations. Documentation of every ethical violation committed in the name of medical advancement. Why are you giving this to me? Because you're right. Someone needs to be a witness. But if I release this, they shut down the facility, and all the researchers disappear into classified projects scattered across the country. The work continues just with less oversight and no accountability. If I release it, you're already out, already documented as a security risk. You have nothing left to lose except your freedom, and they're already planning to monitor you for the rest of your life anyway. Might as well make it count. He gave me directions to a journalist he'd contacted, someone with experience in classified disclosures, someone who knew how to protect sources. There's still time to stop the collection tonight, he said. But once you drive away, you can never come back. Never contact me again. Never acknowledge we had this conversation. I took the hard drive. I drove. The story broke 6 weeks later in the Washington Post. Secret military program harvests DNA from unknown primate species for medical research. Ran above the fold, with photographs from the hard drive, images of darted subjects lying unconscious while collection teams worked, of laboratory facilities hidden in national forests, of genetic sequences that showed clear evidence of human-level cognitive capability. The response was immediate and overwhelming. Congressional hearings were demanded. Animal rights organizations filed lawsuits. Bioethics committees issued statements condemning the research. The NIH director resigned. 3 military contractors were suspended pending investigation. The project was shut down within a week, but not discontinued, just moved, reclassified under even deeper security protocols, distributed across multiple facilities where oversight would be harder and public attention couldn't penetrate. I testified before Congress twice, always in closed sessions. My identity protected, but my credibility constantly questioned. The military's lawyers argued I'd stolen classified information, violated security protocols, endangered national security. My lawyers argued I'd exposed human rights violations against a sentient species. The debate centered on one question. Were they people? The genetic evidence said yes. The cognitive markers said yes. But without living subjects to study, without behavioral observations, without definitive proof of language or culture or symbolic thought, the question remained technically unanswered. And in the absence of definitive answers, national security interests prevailed. The Forest Service designated the known territories as sensitive biological research areas, closed to public access, monitored by enforcement that looked more military than civilian. The official explanation was protection of an endangered species. The reality was containment of a resource too valuable to abandon and too controversial to acknowledge publicly. I lost my career. My name became associated with conspiracy theories and classified research scandals. Universities wouldn't hire me. Research institutions avoided me. I took a job teaching high school biology in Eugene, Oregon, where the principal was willing to overlook my history in exchange for someone who could actually explain genetics to teenagers. Dr. Ko died 8 months after the story broke. Heart attack, officially. He was 64. His death came 3 days before he was scheduled to testify about the full scope of the research program. I attended his funeral and said nothing to anyone about what he'd done, the risk he'd taken to ensure the truth survived. The hard drive is in a safe deposit box, with instructions for its release to multiple journalists if anything happens to me. Insurance of sorts, though I'm not sure what it actually protects. Sometimes I wonder if we made things worse. If exposing the project just drove the research deeper underground, made the subjects more vulnerable because now their value is known. If silence would have been kinder, but I think about those genetic sequences, about juveniles being darted and biopsied for medical treatments they'd never benefit from, about a species that survived for hundreds of thousands of years by hiding from us, now tracked and tagged and harvested because we decided their biology could serve our purposes. I think about the dream I had that last night at the facility. The presence in the forest, watching, understanding, making choices about when to reveal themselves and when to remain hidden. They're still out there, still avoiding us, still surviving in the spaces we haven't yet completely conquered. But now they know something they didn't before. We found them. We took from them. And we'll come back if we need to. The greatest tragedy isn't what we discovered about their biology. It's what we revealed about our own.